Pancreas and its secretion. This is important topic for the B.Sc classes in the animal physiology according to syllabus of Secretin University, and this topic is presented before you by the Seth Ranglal Kothari Government College, Rajasthan. Pancreatic secretion. Pancreas, which lies parallel to the and beneath to the stomach, is a compound gland with most of its internal structure similar to that of a salivary gland. The pancreatic digestive enzymes are secreted by pancreatic acids, and large volume of sodium bicarbonates are secreted by the small ductules and large duct leading to from the acini. The combined product of the enzymes and the sodium bicarbonates that flow through the long pancreatic duct that normally joins the hepatic duct immediately before it empties into the duodenum through the papilla of Vater by the sphincter of Oddi. Pancreatic juice secreted most abundantly in response to the presence of chyme in the upper portion of the small intestine, and the characteristics of the pancreatic juice are determined by some extent by types of food in the chyme. The pancreas also secretes insulin, but this is not secreted by the same pancreatic tissue that secretes intestinal pancreatic juices. Instead, insulin is secreted directly into the blood, not into the intestine, by the islet of Langerhans that occurs in the islet patches throughout the pancreatic tissue. And this will be discussed in another chapter. Pancreatic digestive enzymes. The pancreas contains multiple enzymes for digesting all the three major food sources of food: protein, carbohydrates, and lipids, fats. It also contains large quantities of bicarbonate ions which play an important role in the neutralizing of the acid secreted by the stomach when chyme empties into the duodenum. The most important of the pancreatic enzymes for the digesting proteins are the trypsin, chymotrypsin, and carboxypeptidase. By far most abundant of this is trypsin. Trypsin and chymotrypsin splits the whole protein into partially digested proteins, which are peptides and various size, but they do not release individual amino acids. However, carboxypeptidase does split some peptides and proteases into the individual amino acids, thus completing digestion of some proteins all the way to the amino acid state. Pancreatic enzymes for the digesting carbohydrates is a pancreatic amylase, carbohydrate accepts the cellulose to form mostly disaccharides and few trisaccharides. The main enzyme for the fat digestion by the pancreas is the pancreatic lipase, which is capable of hydrolyzing neutral fats into the fatty acid and monoacylglycerols. Cholesterol, which causes hydrolysis of the cholesterol esters and phospholipids, is split into the fatty acid from the phospholipids. When first synthesized in the pancreatic cells, the proteolytic digestive enzymes are in the inactive forms. Trypsin in the trypsinogen form. Chymotrypsinogen is the proactive form of chymotrypsin, an inactive form of carboxypeptide disease, which all are inactive enzymatically. They becomes activated only after they are secreted into the intestinal tract. Trypsinogen activated by an enzyme called enterokinase, which is secreted by the intestinal mucosa when chyme comes in contact with the mucosa. Also, trypsinogen can autocatalytically activated by trypsin that has already been formed from the previously secreted trypsinogen. Chymotrypsinogen is activated by trypsin to form chymotrypsin, and procarboxypeptidase is activated in a similar way. Secretion of the trypsin inhibits prevents the digestion of pancreas itself. It is important that proteolytic enzymes of the pancreatic juice not becomes activated until after they have been secreted into the intestinal lumen, because the trypsin and other enzymes would digest the pancreas itself. Fortunately, the same cells that secrete proteolytic enzymes into the acini of the pancreas, they secrete simultaneously another substance called trypsin inhibitors. These substances are formed in the cytoplasm of the glandular cells, and it prevents the activation of the trypsin both inside the secretory cells and in the acini and ducts of the pancreas. And because it is the trypsin that activates the other pancreatic proteolytic enzymes, trypsin inhibitor prevents the activation of the other as well. When the pancreas becomes severely damaged or when it becomes blocked, large quantities of the pancreatic secretion sometimes becomes pulled into the damaged area of the pancreas. Under these conditions, the effect of the trypsin inhibitor is often overwhelmed, in which case the pancreatic secretions rapidly becomes activated and can literally digest the entire pancreas within the few hours, giving rise to the condition that called acute pancreatitis. This sometimes is lethal because of accompanying circulatory shocks. Even if not lethal, it usually leads to the subsequent life of the pancreatic insufficiency. Although the enzymes of the pancreatic juice are secreted entirely by the acini of the pancreatic glands, the other two important components of the pancreatic juice, bicarbonate ions and water, are secreted mainly by the epithelial cells of the ductules and ducts that leads from the acini. When the pancreas is stimulated to secrete copious quantities of the pancreatic juice, the bicarbonate ion concentration can rise to as high as 145 millimole equal per liter. A value is about 5 times that is bicarbonate ion in the plasma. This provides a large quantity of the alkali in the pancreatic juice that serve to neutralize the hydrochloric acid emptied into the duodenum from the stomach. The basic steps in the cellular mechanism of the secretion: sodium bicarbonate solution into the pancreatic ductules and ducts. First, carbon dioxide diffuses interior of the cell from the blood, and under the influence of the carbonic anhydrase, They combine with the water and form carbonic acid H2CO3. The carbonic acid in turn dissociates into bicarbonate ion and hydrogen ion H plus and SO3 minus. Then these bicarbonate ions are actively transported in association with the sodium ions through the luminal border of the cells into the lumen of the ducts where they combine and form sodium bicarbonates. The hydrogen ions formed by the dissociation of the carbonic acid inside the cell are exchanged by the sodium ions through the blood borders of the cell by a secondary active transport process, and this supplies the sodium ion that are transported through the luminal border into the pancreatic duct lumen to provide electrical neutrality for the secreted bicarbonate ions. The overall movement of the sodium and bicarbonate ions from the blood into the duct lumen, and it creates an osmotic pressure gradient that causes osmosis of the water also into the pancreatic duct, thus forming an almost completely isoosmotic bicarbonate solution. Here is a figure which represents how the transportation of the sodium ion, hydrogen ion, and bicarbonate ion takes place. Initially, the carbon dioxide from the blood transported into interior of the cell, where this carbon dioxide react with the water in the presence of carbonic anhydrase, and it forms carbonic acid H2CO3, which at once undergoes dissociation into H plus and SO3 minus ion. These SO3 minus ion are secreted into the lumen of the duct, and H plus ion are secondary actively transported into the blood with the taking sodium ion in place of the hydrogen ion. These sodium ions are also secreted into the lumen where sodium and bicarbonate ion combine to form sodium bicarbonate, and they neutralizes the acidity provided by the HCl from the stomach. Whereas in the blood, the ionic equilibrium is established by the exchange of Na plus to hydrogen ion. Regulation of pancreatic. So there are few stimuli that cause pancreatic secretion. These are three basic stimuli are important in causing the pancreatic secretion. First, acetylcholine. Second, cholecystokinin. And third is secretin. Acetylcholine, which is released from the parasympathetic vagus nerve endings and forms other cholinergic nerves in enteric nervous system. Cholecystokinin, which is secreted by duodenal upper jejunum mucosa when food is entered the small intestine, and the secretin, which is also secreted by the duodenal and jejunum mucosa when highly acid food enters the small intestine. The first two stimuli, acetylcholine and cholecystokinin, stimulates the acinar cell of the pancreas, causing production of large quantities of the pancreatic digestive juices. But relatively small quantities of the water and electrolytes to go with the enzyme without the water. Most of the enzyme remains temporarily stored in the acini and ducts until more fluid secretion comes along to wash them into the duodenum. Secretin, in contrast to the first two basic stimuli, it stimulates the secretion of the large quantities of the water solution and sodium bicarbonate by the pancreatic ductal epithelium. Multiplicative effects of different stimuli. When all the different stimuli of pancreatic secretion occur at once, the total secretion is far greater than some of the secretion caused by each one separately. Therefore, the various stimuli are said to multiply the potentiate one another. Thus, pancreatic secretion normally results from the combined effect of multiple basic, not the one alone. Phases of pancreatic secretion. Pancreatic secretion occurs in three phases. These phases are as similar as to stimulate the secretion of the stomach or gastric secretion. These are the Syphilic phase, gastric phase, and intestinal phase. And their characteristics: Syphilic and gastric phases. During the Syphilic phase of pancreatic secretion, the same nervous signals from the brain that cause the secretion of the stomach also cause acetylcholine release by the vagal nerve ending in the pancreas. These cause moderate amount of enzyme to be secreted into the pancreatic acini, accounting for about 20% of the total secretion of the pancreatic enzyme after a meal. But little of the secretion flows immediately through the pancreatic ducts into the intestine because only small amount of water and electrolytes are secreted along with the enzymes. During the gastric phase, the nervous stimulation of the enzyme secretion continues, accounting for another 5% to 10% of the pancreatic enzyme secreted after a meal. But again, only a small amount reacts the duodenum because the continued lack of significant fluid secretion. Second phase is the intestinal phase. After chyme leaves the stomach and enters the small intestine, pancreatic secretion becomes copious mainly in response to the hormone secretin. That secretin stimulates the secretion of copious quantities of the bicarbonate ions and neutralizing the acidity of the stomach chyme. Secretin is a polypeptide. It containing 27 amino acids with molecular weight of 3400 present in an inactive form. Prosecretin is so-called S cells in the mucosa of the duodenum and jejunum. When acid chyme with the pH less than 4.5 to 5.0 enters the duodenum from the stomach, it causes duodenal mucosal release and activation of secretin, which is then absorbed into the food. The one truly potent constituents of the chyme that cause the secretin release into the hydrochloric acid from the stomach. Secretin in turn cause the pancreas to secrete large quantities of the fluid containing a high concentration of the bicarbonate ions up to 145 MEq per liter, but a low concentration of chloride ions. The secretin mechanism is especially important for two reasons. First, secretin begins to be released from the mucosa of the small intestine when the pH of the duodenal content falls below to 4.5 to 5.0, and it releases increase greatly as the pH falls to 3.0. This immediately causes the copious secretion of the pancreatic juice containing sodium bicarbonate, abundant amount of sodium. The net result is then expressed by this equation: HCl plus sodium bicarbonate, NaCl plus H2CO3. Then carbonic acid immediately dissociates into the carbon dioxide and water. The carbon dioxide is absorbed into the blood, and this carbon dioxide is expired through the lungs, thus leaving a neutral solution of the sodium chloride in the duodenum. In this way, the acid content emptied into the duodenum from the stomach becomes neutralized so that the further peptic digestive activity by the gastric juice in the duodenum is immediately blocked. Because the mucosa of the small intestine cannot withstand the digestive action of the acid gastric juice. This is an essential protective mechanism to prevent the development of duodenal ulcers, as is discussed in another chapter. Bicarbonate ions Secretion by the pancreatic provides an appropriate pH for the action of pancreatic digestive enzymes, which function optimally in a slightly alkaline or neutral medium at a pH of 7.0 to 8.0. Fortunately, the pH of the sodium bicarbonate secretion averages 8.0. Cholecystokinin concentration to control the digestive enzyme secretion by the pancreas. The presence of food in the upper small intestine also causes a second hormone. Cholecystokinin is a polypeptide containing 33 amino acids to be released from the yet another group of cells, the eye cells, in the mucosa of the duodenum and upper jejunum. This release the cholecystokinin result especially from the presence of proteases and peptones, product of partial digestion of the protein, and long chain fatty acids in the chyme coming from the stomach. Cholecystokinin, like the secretin, it passes by the way of the blood to the pancreas, but instead of causing sodium bicarbonate secretion, it causes mainly secretion of much more pancreatic digestive enzymes by the acinar cells, and this effect is similar to that caused by the vagal stimulation but even more pronounced, accompanying for 70 to 80% of the total secretion of the pancreatic digestive enzymes after a meal. The difference between the pancreatic stimulation and effect of secretin and cholecystokinin are shown in next figure, which demonstrate first intense sodium bicarbonate secretion is stimulated by the secretin, a dual fact response to soft a fat, and a third instance digestive secretion when peptones enters the duodenum stimulated by the cholecystokinin. This diagram explains how secretion of the pancreas are regulated. When acid from the stomach releases secretin from the wall of the duodenum, and amino acid cause release of the cholecystokinin. Secretin and cholecystokinin absorbed into the bloodstream. They reaches up to the pancreatic cells and stimulate them to secrete secretin, which causes copious secretion of pancreatic fluid and bicarbonate, whereas cholecystokinin causes secretion of enzymes. The same work has also been done by vagus stimulation, which releases enzymes into the acini, but this stimulus is lighter than the stimulus provided to secrete the enzymes by the cholecystokinin. Thank you for watching this video. Have a good day.